I didn't say shh on that side.
Hello, everyone. I'm your host, Jenny Morganwick, and welcome to another edition of JAX Live. Joining us today is Dr. Dave Serez, who's a professor with the Jackson Laboratory, and also we have Dr. Jennifer Dwyer, who is a postdoc with Dave's group. Both of them are doing different types of research in the autoimmune world with focus on type 1 diabetes and genetics. So today we will take the time to welcome them and get to know them in a bit and hear about their research. So welcome to both of you. Thank you for joining us.
Thank you for having us.
Thank you for having us.
So in today's interview, we're going to get to know you both a bit better, hear about your research interests a bit more, and talk about, genetics and type 1 diabetes specifically. But before we begin, I want to let the audience know, be sure to use the comment box on whichever platform that you're joining us on today. Ask us a question, send us a comment, let us know where you're tuning in from. And we'll do our best to get through your questions throughout the interview today. And we'll try to have some in the question and answer session at the end of the interview time. So let's get into this conversation. So Dave, you've been with JAX. We were talking offline earlier. 35 years, is that correct? Am I right on that?
Now going on 36.
36. And you've been
not quite, but we're on the way.
So you've been with JAX for a long time. Can you tell us a bit about your research interests and how it's been at JAX being a professor?
Well, okay, maybe I should start with how I ended up at JAX. Okay. I had a fantastic high school biology teacher. And so my sophomore year in high school, which is more years than I want to talk about now, but I knew right then that I wanted to go into biology. So I was a biology major in college. And then one of the courses that I had to take, obviously, was microbiology. And I absolutely fell in love with bugs. Okay, and I'm still in love with bugs. I don't like the one bug that's causing us our problems right now, but anyway, I generally am quite interested in viruses and bacteria. Then I got interested in basically how immune responses are generated to keep those bugs in check. That subsequently led to an interest as well. How does the immune system know to respond to bugs, but leave your body's own tissues alone? And that's when I figured if I could understand an aspect of autoimmunity, that might be able to help answer that question. And then I had the fortuitousness of actually meeting Dr. Ed Leiter, who is now a retired professor here from JAX. And Ed offered me a job to come in and help him because he was importing the NOD mouse, which is a model for type 1 diabetes, which has been feeding my family for the last 35, almost 36 years. And Ed would be the first one to tell you that he was more of a cell biologist and an immunologist, and he needed somebody with a background in immunology to help him do the initial analysis of these mice. So the NOD mouse and I arrived on campus in the same week of 1985. My first day.
Wow.
That's
cool.
My first day at the Jackson Laboratory was May 13th, 1985, a day that will live in infamy, at least in my mind. Anyway, if you told me then I would be, 35 plus years later, I'd be a full professor sitting here, I'd say that you were absolutely insane. But it's been a fun ride. It really has been. And it's been a fun ride because I've actually got to work with great people. My motto is hire people who are a lot smarter than you are, get them into your lab, people like Jen, and then get the hell out of their way. And I've been fortunate that I've had a series of good postdoctoral fellows over the years. And type 1 diabetes has been called the geneticist's nightmare. There's probably over 60 genetic loci that contribute to the disease process. And when you start to dig into those individual loci, and this is true for both the NOD mouse and humans, by the way, there's more than one gene within most of these loci. You're probably dealing with 100 or more genes that are contributing to disease. And trying to dissect that has been it's been challenging, but it's also been a bit of a joy. And the question is, I don't think these are inherently diabetes genes per se. Because if you think about it, we only discovered insulin in the 1920s, okay, in order to treat type 1 patients. And that isn't enough time for any genetic change. My mother was born in the 1920s, and she is still fortunately with us. But without insulin treatment, type 1 diabetes is a lethal disease. So you would have thought that if these were inherently diabetes genes, they would have been eliminated by natural selection a long, long time ago. And that clearly has not happened. So what I think these genes originally evolved to do is to give you very potent, getting back to bugs, is giving you very potent immune responses to pathogens that we no longer commonly encounter. Okay, but now, at least in aseptically privileged Western societies, but now that we're no longer challenged by these bugs to the extent that we once were, I think we're seeing these genes being able to exert a dark side, and that's contributing to autoimmunity. There's a huge amount of genetic overlap between the genetic basis for multiple autoimmune diseases. A lot of type 1 patients, unfortunately, also do suffer from other autoimmune pathologies. So that's really what I think is a driving challenge of the field these days.
So Jen just let me know that she had to go grab a cord for her laptop because she was going to lose us. So we'll come back. We'll have her join us the interview in a little bit.
She's back.
You're back.
That was dropping fast. It was 10 and 7 and 5 and 4 and 3. So yikes.
Well, that was a quick run.
Sorry, I apologize, Dave.
So thank you, Dave. That was a great insight to, how you got to where you are. Great, high level overview of the complications of type 1 diabetes and genetics. I think a lot of people sometimes expect just 1 or 2 genes to contribute to a disease, and they don't realize how complicated it can get. And that's why it's so hard to treat.
Most common diseases are actually polygenic in nature.
And that makes sense. We're a complicated being. So Jen, let's flip to you for a second. Let's get the audience warmed up to who you are. So tell us a bit more about your journey. What brought you to JAX? What led you to a postdoc with Dave? How his work complemented your interests?
Sure, I'm super happy to share with you all my path to JAX. Mostly because I have a kind of unconventional road as a postdoc. In my family, type 2 diabetes is very common. And so as I was growing up, I always had an interest in physiology and metabolism. And so as I was going through my biology degree in undergrad, I gravitated towards anything that had to do with physiology. And when I went to graduate school, I studied transgenic and knockout mouse models of lipid metabolism. And admittedly, I was headed towards a career in type 2 diabetes at the end of graduate school and 2 years into my postdoc. But about 2 years into your postdoc is really when life happens. I was married, I had 2 children, and I said, oh my goodness, I need to take a break and take care of my young family. And so it was a hard decision, but it was something that needed to happen. And so the plan was to take like a 2-year break. Science moves really quickly, and I didn't want to be out for too long. So, but about 6 months into my break, my son was diagnosed with type 1 diabetes, and he is 6 years old at the time. Now, I was super familiar with all the metabolic complications of uncontrolled type 1 diabetes, and so I had an advantage there. But now I'm on a break from science. I want to go back to science. So I had a tough decision to make, which was to either follow my career and get on the horse back to, being a type 2 diabetes researcher, or do I change fields almost entirely and move towards immunology and type 1 diabetes, because type 1 diabetes is an autoimmune disease, as Dave mentioned, but type 2 diabetes is more metabolic in nature. So I started reading about type 1 diabetes while I was at home and becoming familiar with the different areas of research. First of all, there's stem cells and generating beta cells to replace the ones that get destroyed by the autoimmune attack. But once you can be successful at generating beta cells and can implant them, there's challenges that those cells face. First of all is to avoid graft versus host disease. And the second is, how do you protect those beta cells from the autoimmune response against particularly specifically beta cells that exists in type 1 diabetes? And So I had a decision to make, and I come from a family of engineers. Dave comes from a family of scientists and chemists, and my family's engineers. And so I've always liked to figure out how things work. And I remembered a story from when I was a kid. My grandfather was helping me fix my bicycle once. And of course, we had the bicycle upside down in the basement with the parts everywhere, and we fixed it and got it back together. And as we finally got it, the pedals spinning, the chain spinning, the wheels spinning, and everything's clicking away. And of course, I just wanted to ride my bike. My grandfather said to me, and he was an electrical engineer, he said, wait, watch it spinning and clicking, and how all the parts move together, and they have to work. Everybody has to do the right job to make the whole thing work together. And so you have to understand how the bike works and how each and every part and gear and chain works together to make the bike work. And so as I read about type 1 diabetes, I felt like there were mysteries in the autoimmune response in type 1 diabetes. Like, why does it develop in the first place? What's the part of the immune system that is broken in type 1 diabetes? And so I started researching labs that study this, and I have a background in genetics. And I was immediately drawn to Dave's lab. And as a graduate student in genetics, I was super familiar with the high quality standards at JAX. And I reached out to Dave for an informational interview, and we hit it off. And I'd encourage all postdocs who are thinking of where their career is going to just talk to people as much as you can. We were able to secure some funding to get me back into a career reentry grant, and the rest is history. So that's my path to JAX.
Well, that's great. I know JAX is happy to have you. So Dave, I want to shift focus and talk about a publication that you came out with in 2018. And what I like about this publication is I feel like it really kind of gets to the heart of the question of, the criticism around mouse models not adequately recapitulating the human condition or disease. So a lot of people say, well, what's the point of using them? But in your 2018 publication in diabetes, you covered this very specific topic. So essentially you point out that the non-mouse model of type 1 diabetes, while it does have some shortcomings, one of those being utilization and efficacy studies for potential therapeutics. So can you talk about the changes that your group made to this model, the NOD model, and can you highlight some of your findings from that paper?
Yeah, sure. So first of all, I'd point out that over the years, my group has actually made over 400 genetically engineered variants of the NOD, all with a focus of actually trying to develop means that you could eventually either have ways to develop therapeutics that will either prevent the onset of type 1 diabetes in humans deemed to be at high future risk. And we in the type 1 community now have about over a 90% capacity to identify humans at future risk. But the question is, what can you do for them? Right now, nothing. I mean, things are being tried, but there's nothing that has been proven yet. Or is there anything you can do for once a person has already developed type 1? Is there any way that you can treat and reverse disease? And that's going to be even a harder challenge. It's my hope in my career that we at least can achieve the former, be able to identify humans at risk and be able to do something for them. So this paper that you're talking about in 2018, even though type 1 diabetes is a highly polygenic disease, I mentioned a few minutes ago, probably over 100 genes that are ultimately contributing to the disease, the major contributors to type 1 diabetes and other autoimmune diseases are what we call major histocompatibility complex, MHC, and that's designated HLA for human leukocyte antigens in people. And just as an aside, when you hear somebody needs a heart transplant, kidney transplant, what have you, and they're looking for a quote unquote match, what they're looking for is a match at the MHC or HLA. And just as an aside, the MHC was actually discovered and defined by Dr. George Snell here at the Jackson Laboratory. So basically all tissue transplantation is made possible because of Dr. Snell's work. And Dr. Snell was actually one of my early mentors here at Jackson. It was very rewarding for me personally to get his insights. But it turns out these MHC genes also play an important role in driving various autoimmune responses. So what we did is we used a technology called CRISPR, which actually has been in the popular press as well. And what we were able to do is we were able to use CRISPR to knock out NOD MHC genes, the mouse MHC genes, and then we replace them with human MHC genes. And it turns out in these mice now develop diabetes, still develop type 1 diabetes, but they develop diabetes that's mediated by human HLA genes rather than the mouse MHC. So the T cell responses that are actually causing the disease in these mice are of pathological relevance to a large prevalence, over 60% of type 1 patients. So if you can find ways to attenuate the autoreactive T cells that are causing type 1 diabetes in what we call these HLA humanized mice, that intervention could also potentially be of relevance to type 1 patients or people that are at future risk for developing type 1.
So we actually have a great question from the audience. In the past, we've had a couple live streams that have talked about antibody therapeutic development. And I know that the antibodies as treatments are pretty big in the world of immunology. So someone from the audience wants to know if you're working with antibodies as possible drug compounds, is the FcRn functionality in these mice intact?
Yes. Okay. And the reason for that, it's a very good question, by the way. And the reason it's a good question is a first generation, there was an approach that you could knock out the expression of mouse MHC molecules, MHC class 1 molecules, by removing an expression of another molecule called beta-2 microglobulin. So the early approach, this was done back in the 90s, for eliminating expression of mouse MHC class 1 molecules was to knock out this beta-2 microglobulin. Well, it turns out that beta-2 microglobulin is also required for the expression of this molecule called FcRn that Jen is asking about. And FcRn is also expressed on the surface of blood vessels. And it's a very important molecule that prevents the catabolism or breakdown of antibodies. So beta-2 microglobulin mice, yeah, they don't have mouse MHC class 1, but they also do not have FcRn. So if you put an antibody into these mice, these beta-2 microglobulin deficient mice, that antibody is rapidly degraded and obviously can't do whatever you want that antibody to do. So if you're looking at antibody-based or potential antibody-based interventions for type 1 diabetes or any other disease for that matter, it wouldn't work in the beta-2 microglobulin deficient version of mice. We use the CRISPR to directly ablate the expression of the mouse MHC molecules. So as a result, they retain FcRn. And we actually tested in the paper that Jen is talking about. We said, okay, if we put antibodies into these mice, are those antibodies maintained at high levels for a significant period of time? And the answer, fortunately, was yes. So you could test antibody-based therapies in these next generation mice that we've made that would not have been possible in previous generation mice.
Oh, that's a fantastic improvement.
Yeah. Hey, I see a question here on the side. It's interesting. I've got a shout out from California, Boston, Indiana, and a former classmate from Dave's high school biology class. I'd like to know who that is.
Let's see if we can get our magic behind the scenes to see who that is that's saying hi to us. Okay. So while we're waiting for that answer, Jen, is there any new and recent exciting discoveries or results that you want to share with our audience with your work?
Yeah, actually, there's just very quickly, there's one that I'd like to share. It's one component of type 1 diabetes development is the circulating cells that are autoreactive, they're T cells. And usually in a normal person, they're deleted during thymic the development of the T cells in the thymus. And Dave's lab, or type 1 diabetes, there's a failure to delete these cells and inhibit the activity of any ones that remain. So Dave's lab and the postdoc before me fine mapped this gene, or region of the genome that carries this trait, and I isolated a gene that's contributing to this phenotype. And when they knocked out the gene directly in the NOD mice using CRISPR-Cas9, they found that removal of the gene actually accelerated disease. And so I'm really fortunate now to be able to study the transgenic overexpression of this gene, which is actually resulting in a striking protection from type 1 diabetes. So my project is to isolate the mechanisms that are responsible for that.
That's excellent.
And she's doing a fantastic job at that, by the way.
It sounds like it. So we've got time for one more question. And Dave, this is one that I want to direct to you. You've been with JAX for a while now, and you've been, putting out excellent research this whole time. I want to hear from you. What is your proudest moment as a scientist, or your biggest breakthrough that you want to share with our audience?
Okay, I've got two answers to that. I guess from a personal point of view, probably the most ingratiating moment for me is when I was selected to win the Grodsky Award from the Juvenile Diabetes Foundation for outstanding contributions to diabetes research. And what made that particularly special to me, it was not just for a particular paper that we did or a particular discovery, but it was for the body of work throughout my career. And that meant a lot. The other thing that I'm most proud of is I've had a series of postdocs that have come through my lab, and then they've gone on to get their own independent academic positions, and they also remain in diabetes research. And I always consider these to be my scientific offspring. And so I think that those would probably be two.
Well, I know a couple of your scientific offspring personally, and they are excellent scientists. So you're doing a great job. So I want to thank you both for joining us today. It's been a great conversation. I wish I could talk to you guys more about this for, a couple more hours. I'm fascinated by the work that you guys are doing. But unfortunately, we're out of time. So if we didn't get to your questions in the audience, we'll be sure to follow up with you. Again, thank you both for joining us. Hope you have a great day, and join us next time, same place for your next live stream. Thank you.